Hello, everyone, and welcome to today's webinar, Current and Future Directions of the NIH Brain Initiative. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the continuing education window at the bottom of your screen to obtain your credits. I'd like to now welcome our speakers, Dr. John Ngai, Director of the NIH's Brain Research through Advancing Innovative Neurotechnologies, or BRAIN Initiative, and Dr. Mohammad Ghassemi, Assistant Professor of Computer Science and Engineering at Michigan State University. Dr. Ngai and Dr. Ghassemi, you may now begin your presentation.
Hi, first of all, I'd like to thank the organizers for this invitation to tell you a bit about the BRAIN Initiative and our current and future directions, which hopefully will serve as a nice introduction and backdrop for the rest of today's exciting talks. So the mission of the Brain Research through Advancing Innovative Neurotechnologies, or BRAIN Initiative, is to revolutionize our understanding of the human brain by accelerating the development and application of innovative technologies. This initiative was announced by the White House in 2013 with the first awards in 2014 and represents a partnership between 5 U.S. federal agencies as well as private foundations. Now, the NIH efforts have been guided by 2 strategic plans, the so-called BRAIN 2025 report that was issued in 2014, as well as a revisit of the strategic plan in 2019 as issued in the BRAIN 2.0 reports. Now, the goal of the NIH Brain Initiative is to develop and apply new tools for understanding how neural circuits underlie complex behaviors in both health and disease, arguably the central theme of today's symposium. To do so, we need to leverage the technological innovations to enable new discoveries about neural circuit function, to use these discoveries as a foundation for new therapeutic strategies for human brain disorders, and very importantly, to disseminate and democratize technologies for basic discovery and clinical applications. Now, I mentioned the BRAIN 2.0 reports, this revisit of the strategic plan, and I just wanted to summarize for you some major points that were brought up in both the BRAIN 2.0 neuroscience report as well as the neuroethics report. The first was to stay on the productive path that was already underway for the BRAIN Initiative. In other words, keep on doing what's working well, to continue supporting technology development and targeted study of circuit components, which you'll hear a lot about today. The second was to balance individual and investigator research with team science. There's clearly a place for both. The beauty and I think success of NIH-funded research is that we ask investigators to come in with their best ideas and to pursue exciting hypothesis-driven research. At the same time, there are some projects, which we'll talk a bit about today, that really do require a team approach. Some of it hypothesis-driven, but some of it driven toward building tools that allow us to ask and answer better and more precise questions. The third point I'd like to emphasize here is the importance of enhancing the integration of neuroscience and neuroethics. This question of what are the impacts of our work, how will that impact broader issues in society, and not to consider these just at the tail end of our studies, but really to bake it in from the very beginning. So we really do understand what we're getting into and that we can navigate space, which at some points can be quite fraught, especially as it involves the human brain and human behaviors. And then finally, the last point that I'd like to emphasize here is this suggestion to devote ample resources towards scaling up large projects that could transform the way we conduct neuroscience research for generations to come. Now, you can also get more details about this plan at the link that I show here at the bottom of the slide. So I'd like to spend a little bit of time here today to describe to you these so-called Brain 2.0 transformative projects that we are embarking on for the next phase of the Brain Initiative, the next 5+ years. And hopefully you can see how this will impact the work that's going to be discussed today and beyond, and then I'll tie it in with the subject of today's talks. So we chose to pursue 3 large projects, these so-called transformative projects that we do believe will transform the way we conduct neuroscience research in the future. The first is the phase 3 of the brain cell census project. This is to build literally a parts list of the brain. Right now we are in phase 2, the so-called Brain Initiative Cell Census Network, or BICCN, where the focus has mainly been on the mouse brain, but with some work on non-human primate and human brains. And for phase 3, we will be moving the emphasis primarily, but not exclusively, to generate comprehensive cell atlases of human brains. Now, a series of RFAs were just released. They're shown right here, with receipt dates coming in November 2021. So let me just give you, in turn, a bit of background for each of these 3 transformative projects, starting here with this Brain Initiative Cell Atlas Network, and I'll move on to the other ones as we go along. I should also mention that Congress has allocated to us specific funds to allow us to launch this transformative project starting in next year. So the idea and motivation behind the cell census work is that we need a parts list and a wiring diagram for the brain, and a way to interrogate and modulate brain circuits. So the 3 projects I'll tell you about today touch on each of these 3 areas: the parts list, the wiring diagram, and a way to interrogate and modulate. So in order to understand how this marvelous biological computer works, we really need to understand what are its components, how many do we have of each, what are their properties, and very importantly, how do they communicate with each other? How do they wire in with each other? Now, this has been a century-old problem dating back to the days of Ramon y Cajal. But what we need right now is a systematic way to identify and classify and characterize these hundreds, if not thousands, of different cell types that comprise the mammalian brain. So this effort started back in 2014 in a pilot phase of the Cell Census Project, which was referred to as the Brain Initiative Cell Census Consortium. And the idea behind the Brain Initiative Cell Census Consortium, or BICCC, was to identify and validate scalable technologies that could be deployed to result in a comprehensive brain cell atlas based on the multiple properties exhibited by individual cell types. So I was fortunate to be a PI in that effort way back in 2014. And then the next phase of this project was the Brain Initiative Cell Census Network, which was a scale-up of the effort based on the lessons learned in those pilot studies. And so the goal of the Brain Initiative Cell Census Network at BICCN was to revolutionize the ability to classify brain cell types based on an integrated analysis of their molecular, anatomical, and physiological properties. And this was a bold ambition, but I think we have made quite a bit of progress here. So let me just describe to you what the BICCN, or Brain Initiative Cell Census Network, is in terms of organization, and hopefully you can see how this will provide a foundation for the much more challenging endeavor of scaling this project to characterizing cell types in the human brain. So the BICCN is a multi-project consortium or network. It's centered around 3 large U19 production projects shown here at the top of the slide, focused again on the mouse brain, together with a series of smaller but quite substantial UO1 projects aimed mainly at identifying connectivity properties among those cell types that are identified. To the right in red are a series of UO1 projects that are focused on characterizing cell types in human and non-human primate brains. And on the left are a series of R01 type projects aimed at developing improved scalable tools and assays to accelerate the entire endeavor. And actually, this slide is a bit out of date. There are more projects ongoing that are listed right here. Now, the efforts among the BICCN are coordinated through this Brain Cell Data Coordination Center, or BCDC, which coordinates not only the efforts among the entire network, but also helps organize the data coming out of this network into a series of data archives shown toward the bottom in light blue, as well as data integration and analysis projects that are related to that, all using data standards that are currently under development. Now, the ultimate goal here is to disseminate this information out to the general scientific community, shown here schematically toward the bottom. So very importantly is this structure to coordinate the activities. And here you can see in the middle of the slide, the Brain Cell Data Center, or BCDC, that coordinates and directs traffic to make sure that the data are winding up in the brain data archives. Here, the three main data archives for the BICCN are the NEMO archive for transcriptomic and epigenomic information, the Brain Imaging Library for microscopy and imaging information, and the more recently stood up DANDY archive for neurophysiology data. And on the right, we're in the process of developing tools that can be used by other users outside this group to both mine and analyze and interpret the information that's held in these archives. So a very, very important component is how we get the data out there and make it accessible for all to use. So just as an example of where this study has gotten to so far, as a test case to see whether this network could actually work collaboratively and effectively to generate a multimodal atlas, in other words, an atlas of cell types based on multiple modalities, there was a decision made early on to focus on one part of the cortex, in this case, the primary motor cortex, in mouse, marmosets, and humans, using a variety of approaches, which are shown here on the second level of this diagram, various iterations of single cell or single nucleus RNA sequencing or transcriptomics, single nucleus methyl C sequencing, single nucleus ATAC-seq to identify regions of open chromatin, to give us a comprehensive outlook of what cell types might look like based on the molecular information, MERFISH, or high throughput in situ hybridizations to gain spatial information about molecularly identified cells, a technique known as PATCH-seq, which now allows us to gain information on single cells, not only about their transcriptomics, but also about their morphology, location, as well as physiology, so truly integrating across multiple modalities. Ways of tying in all this molecular information with the connectivity patterns of these cells, and a technique that was been named epi-retroseq, as well as other single cell morphology techniques to again characterize the long-range connectivity patterns of the molecularly identified cell types. So this has resulted in a multimodal analysis of cell types in the primary motor cortex across mouse, marmosets, and humans. And what we've gained here are the organizational principles underlying the diversification of brain cell types, here using as an example the primary motor cortex. And what we see, as from many take-home lessons, one I'd like to highlight here is the remarkable conservation of cell types between mouse, marmoset, and humans. And this really does set the stage for scaling up the entire endeavor to generate a comprehensive brain cell atlas from humans. Now, this is really a great example, I think, of the power of team science. There were over 250 researchers involved in this endeavor, with 89 affiliations spanning over 45 institutions, hailing from 3 different continents. So this was a great team effort, a highly collaborative effort. There was as much knowledge gained about science as there was about the sociology of science here, and we are really quite excited to see these advances, both in the science as well as how we do science, toward this much larger scale project where we will be attacking the entire human brain as a resource, not only for understanding how the human brain works, but also as a resource for devising future human therapies for circuit disorders. So the next project I'd like to tell you about is the so-called Armamentarium project. Here is a project to organize resources for brain cell type access and manipulation across multiple species. So the goal here is to initially gain experimental access to brain cell types in species that may not have such ease of genetic access through the germline. So in non-human primate brains and human cells and tissue. Many of these tools will involve viral vector technologies, but also there are non-viral vector technologies in development as well. The long-term goal here, in addition to generating better tools for interrogating and modulating neural circuits, one of the goals that I listed a few slides ago, is to develop new therapeutic strategies for human brain disorders using the latest developments in gene therapies. And this is a ways off, but we really do need to start by having ways of accessing these cell types somatically. Now, we've already issued an RFA for this. We've already had one round of applications come in with a second receipt date coming later this fall. And we also have another RFA, the second one listed here, for standing up production facilities in partnership with these initial technology development projects. So let me just lay out for you a bit of groundwork of why we think this is an exciting way to go. And again, this is another project that for which we receive congressionally directed funds to jumpstart the effort in actually this fiscal year. So the goal here is to develop new technologies for cell type access somatically. One approach for this is to identify enhancers that will direct cell type specific expression in adeno-associated virus vectors. And here's just a highlight of a recent publication from Basilka Tasik and her colleagues at the Allen Institute, where they use single-cell ATAC-seq to identify conserved regions of accessible chromatin associated with specific cell types in the mouse brain. And then they screen these sequences for enhancer activity to drive reporter gene expression in specific brain cell types as delivered through recombinant adeno-associated virus vectors. And they showed that by using a combination of a combinatorial approach where they can mix and match different vectors with different specificities as driven by these enhancers, they can provide a very flexible and facile access to brain cell types that complement germline methods. Now, this was initially developed using the mouse as a model, but in associated studies from Jonathan Ting's group, also at the Allen Institute, they could show that some of these enhancers were also active in non-human primate brains as well as in human brain tissues. So I hope you can see that there is a path not only toward generating better reagents for accessing cell types in experimental models, but now also a path toward thinking about how we might leverage this information to access cell types in humans for the purposes of gene therapy. So this is where we are today, and we're really excited to expand this effort to develop a greater and more extensive toolkit for brain cell access. So the third project I want to tell you about is in a more formative stage. This is the whole brain microconnectivity analysis of the so-called wiring diagram. And the goal here is to develop a connectivity map of an entire mammalian brain, and hopefully brains, at multiple scales, from the macro to the meso to the micro or nano scale. The Brain Initiative, together with the Department of Energy Office of Science, recently hosted a 5-workshop series looking at the state of the art and the challenges and the opportunities for what it would take to prosecute such an effort. We've recently published a workshop series report that's available online at the following DOI. You can also find this at the Brain website. So let me just lay out for you in a few slides what the challenges of stepping up such a project really might be. So here is a summary of a study that was performed by Jeff Lichtman's lab back in 2015, quite a while ago, where they used automated serial sectioning of mouse cortex combined with scanning electron microscopy and a lot of very intensive statistical analysis for 3D reconstruction to reconstruct the cells and their connections within a roughly 1.5 millimeter cubed volume of the mouse cortex. So the entire mouse brain is about 500 to 1,000 times larger than this 1.5 millimeter cube piece of cortex that Lichtman and colleagues reconstructed. So the challenge is how to scale this up to an entire mammalian brain at synapse level resolution. Is that even possible today? Now, the experimental challenge is collecting data from a whole brain. This will require some way of sectioning the brain losslessly. In other words, to section it but not lose all the bits in between, and ultimately being able to put them all back together again accurately. And we want to be able to bridge scales, again, from the macro scale to understand what connects different brain regions to each other, to the meso down to the micro scale, where we want to understand what are the synaptic connections being made locally. So this is a big challenge. It's also a huge data science challenge to conduct the segmentation, reconstruction, and proofreading of such data sets. One whole mouse brain at nanometer resolution is estimated to occupy 1 exabyte of data. That's a humongous data set. A human brain, which is another 1,000 times larger, again, would occupy 1 zettabyte, which if you go on Wikipedia, it will tell you a zettabyte is roughly the annual internet traffic around the entire world. So this is a huge computational problem. But while we're pondering the computational problems, the theoretical problems of trying to put these maps together, experimentally, how do we bridge across these different scales? And I think one way to approach this is to use multiplicity of modalities. So here's a high-level summary of a recent paper from Bobby Kasturi's group, who showed that they could bridge 5 orders of magnitude of scale by using diffusion tensor imaging, micro CT or X-ray tomography, down to EM microscopy, to go from the macro scale down to the nanoscale, where they use this X-ray tomography or micro CT at 1 nanometer resolution to bridge across these different scales. So on the left here, I'm showing the diffusion tensor imaging. In the middle, we show micro CT, which at 1 micron resolution is able to identify and follow and trace individual axons. And then in the upper right is an EM micrograph showing the fine structure of these embedded ultrastructural elements. So we can go from 50 microns at DTI down to nanometer by EM, bridging it with micro CT at 1 micron resolution. And micro CT identifies cellular features and traces, and as well as traces individual myelin axons, as shown in these figures here. And quite nicely, one can perform the imaging using all these 3 modalities on the same sample. So one can start with DTI, step down to micro CT, and then ultimately to EM. And you can see here on the left is a micro CT image with a corresponding EM image on the right. So there really is a great promise for bridging these scales by using a multiplicity of modalities and analyzing, hopefully on the same sample, to piece all this information together. And the advantage of micro CT is that it's scalable. One can scan an entire mouse brain in 7 hours, and which results in about 10 terabytes of data. Okay, so these are the 3 transformative projects. And what I would argue is that together they will provide for us the ground truth to be able to really understand how circuits underlie behavior. And overall, the discoveries and tools being supported by the Brain Initiative are enabling new opportunities for unraveling the circuits and computations underlying behavior. This will allow us to identify the cells and circuits that are involved. And again, provide the ground truth for informing and constraining testable models for how neural circuits drive behavior. And I want to stress the importance of theory in informing experiment, and conversely, the importance of experiment informing theory, which underscores the critical role of collaboration between experimentalists and theorists if we really are truly to understand how these neural circuits underlie behavior. And the long-term goal of the Brain Initiative is to illuminate general principles that can be applied across the domain of the different behaviors that are supported by the brain. So just to kind of come back and think about the topic of today's symposium, I want to just briefly summarize 2 programs in the Brain Initiative that are aimed at understanding neural circuit function using the new tools that are being developed by the Brain Initiative as well as in adjacent fields. And so the first program I'll highlight for you here is this team research-based brain circuits program, or Team BCP. And the motivation here is based on the one of the recommendations from the Brain 2025 report, our strategic plan that was issued in 2014. And the idea here is that rigorous theory, modeling, and statistics are advancing our understanding of complex nonlinear brain functions where human intuition fails. So in other words, we need a strong theoretical basis for asking good questions, for framing our experiments, and for testing those experiments, again, as a two-way iterative process of experiment validating theory and theory informing experiment. Now, in order to have a full integration of theory and experiment, the purpose here of these large team-based projects is to incorporate multiple components that have teams with diverse research expertise spanning both multiple experimental disciplines as well as theoretical disciplines. They can contain up to 5 synergistic research projects and 5 research cores. And again, the theory model-driven approach is central to all of these projects that are funded through the U19 mechanism. And here's just a high-level summary of the U19 projects that are currently underway that are investigating various aspects of circuit function underlying behavior, from behavior to cognition and memory to perception. And you can also see some pretty clever acronyms for these projects, and you'll learn, I think, a bit more about these projects in the hours to come. So these are the large team-based brain circuit program projects. We also have a series of R01s in the so-called Theories, Models, and Methods portfolio, or TMM portfolio. So again, the idea here is to emphasize the importance of theory in driving the experimentation, and also the experimentation being a check on the theory. And literally, the goal here is to develop better theories, better models, and better methods for analyzing this complex data space for the ultimate understanding of the principles that are underlying neural circuit function. So with that, I'm going to hand it over to our next speaker. But before I do that, I need to acknowledge all the great work that's done by the over 100 NIH program staff that are involved in this effort. And in particular, I'd like to call out one person here, Mohammad Ghassemi, who was our next speaker. Now, Mohammad is our NIH Brain Initiative Data Scholar. It's a special program around NIH to bring in a data scholar to look at and to develop a novel way of parsing and understanding the data that we're generating. Mohammad is currently an assistant professor at Michigan State University, and he's been with us for the past year or so as a data scholar. And what Mohammad will tell you about in the next 15 minutes or so is this BrainWorks project that he's been working on. So BrainWorks stands for Brain Initiative Workspace to Organize the Knowledge Space. In a nutshell, this is to use data-driven, information-based techniques to mine the literature, to mine the information out there, to identify topic areas that are currently being supported by brain, and also, very importantly, to identify regions of intersection. And the idea here is to discover in advance what some emerging new areas of discovery might be based on the work that's being done out there today. So this is just a very high-level overview in this graph where the nodes represent research topics supported by brain, and the edges represent research performed at topic intersections. And you can see here in this one hairball plot, a couple of very well represented nodes, for example, in magnetic resonance imaging, in single cell analysis, which is at the core of the brain cell census project I just mentioned to you. Of course, optogenetics is a very powerful tool for understanding neural circuit function, neural pathways, etc. And what we'd like to do is be able to use this kind and other sorts of graph analyses to identify areas of emerging intersections and ones that might help us predict where the next big discoveries might come from. So that's just a very high-level overview. I'll let Mohammad describe to you the project in much more detail and hand it over to him. So over to you, Mohammad.
Hi, I'm Mohammad Ghassemi, and I'm an assistant professor of computer science at Michigan State University. And this year, it's been my pleasure and opportunity to serve as a Data and Technology Advancement National Service Scholar at the NIH. And my presentation today is going to be taking you through BrainWorks. Now, BrainWorks, at its simplest, is a platform with an ambition of converting scientific papers into a knowledge graph. The overall ambition or motivation for this really contains two pieces. The first is, if you think about scientific disciplines like neuroscience, it's actually composed of many smaller subdisciplines. So if you look on the left-hand side of the screen there, you can see we've got social, genetic, cellular, physiological, neurological, and behavioral subdomains of research, all of which together comprise the domain of neuroscience. And what scientists in these areas are doing is they're taking grants, usually from the government, and they're doing excellent work, and the consequence of that work is captured within scientific manuscripts or papers. And our ambition here is to take those collections of papers across those varieties of subdomains and to create a knowledge graph that captures all of the major findings embedded within those papers and across those papers. Now, you might naturally wonder, well, why should we do something like this? Aren't the papers already there? Can't we just read them? And the short answer is, yeah, absolutely, you certainly could read them. But the scientific domain has, especially over the last 20 years or so, grown increasingly complex, and the volume of scientific output has really been on a pretty steep growth curve. On the right-hand side of the screen there, what I have is the number of neuroscience-related articles. That's the y-axis. And along the x-axis, I've got the year. And so there's 2 points I want to draw your attention to in that graph. If you kind of scroll your eyes to where that first arrow is, you can see that what that signifies is if you wanted to kind of keep up with the literature around 1980, you'd have to be reading something on the order of 13 papers a day. And while that's a lot, I mean, you could argue that maybe you could, if you're really a prodigious reader, you could consume all of those. But if you fast forward to today, you have to read something on the order of 70 papers per day if you wanted to just keep up with what's happening in the scientific community. And it doesn't look like that curve is going to be heading down or flattening out anytime soon. So the thought is that a tool like BrainWorks, by turning the scientific papers into a knowledge graph, might allow us to better digest what's happening within these domains of science and choose research directions a little bit more strategically and learn things that otherwise would have been challenging to observe. Now, as I mentioned at the start of the presentation, I'm here at the National Institutes of Health as a Data and Technology Advancements National Service Scholar. And my appointment here has been ongoing for about 8 months now, and will be continuing until the end of the year. The project or delivery of this BrainWorks platform has happened in 5 phases, and I'd like to very quickly highlight what we've already done and also where we're going in the next few minutes. So the first thing that we did was we had a series of conversations at the very early days of this project with experts inside and outside the NIH. Several of those experts, inside and outside, are folks who you will hear from today or have heard from probably in other domains. I wanted to just put all those individuals and their smiling faces here at the beginning to acknowledge that they were extremely helpful in helping us craft that vision that I articulated in the first couple of slides for this platform. So once we had completed that specification of what we wanted the BrainWorks platform to do, we then moved to the next very important stage of this, which was gathering the data that's going to allow us to build that platform. And ultimately what we came up with in that 3 or 4 months intensive data collection period is illustrated here on the slide in front of you. Starting from the left, we collected information on about 2.7 million funded NIH projects from 1980 to 2020. that collectively account for something on the order of $500 billion in investments in the scientific community. And that $500 billion in investment went to something on the order of 5 million scientists embedded at 100,000 institutions. And those 5 million scientists over the period authored something on the order of 3.7 million papers. And those papers cited an additional 6 million papers or so to lead us to a total collection of around 10 million or 9.5 million papers in our collection. Now what we did is, once we had identified all the projects, all of the scientists who were involved in those projects, and all the papers that those scientists had created, and we had added those to our archive, we added 2 other pieces of information. First that we did was we assigned topics to each of the papers. So we can take a paper that's published by some scientists that were funded by NIH and know, for example, that it was about neuroplasticity, or know that it was about magnetic resonance imaging, or a set of other topics. And we also collected any sort of patents that were authored that were influenced or supported in part by the funds from the NIH. Now, with that data collected, what we've been spending the last couple of months doing is generating tools to analyze and visualize this data set that we have collected. And I'd like to take you through in the next 10 minutes or so what some of those preliminary analytic techniques and visualizations of the data look like. And just note here up front that this tool is still evolving, so there'll certainly be more work to come on what I'm about to show you. So at the end of John's presentation, he showed you something that looked a little bit like this. This is a topics graph. And the way that we generated this was we took all of those papers that exist within that 10 million or so paper collection that we spoke about, and we said, hey, which subset of those papers were funded by the BRAIN Initiative from 2014 all the way up to 2020? And then let's do 2 things with those papers. Let's identify what the topics are, and for each of the topics, let's go ahead and put a node on this graph that you see in front of you. And then every time we see within a given paper 2 topics that occur, let's draw an edge between those 2 topics that we see on this map here. And so the general structure that you see in front of you for this topic map communicates those 2 pieces of information. What are the sets of topics that showed up, as well as what are the co-occurrences between the topics, as communicated by the nodes and the edges respectively. Now there's a couple of other pieces of information that this topic graph highlights for us. The first is we've sort of made the text hovering above the nodes larger if that topic was more represented or showed up in the papers more. So you can see magnetic resonance imaging as an example. There's a large piece of text, single cell analysis, optogenetics, and so on. And then if we want to look at an example a little closer to the top of the graph, we can see that there are connections between neural pathways and movement. And so there were studies that have been funded by the BRAIN Initiative at those interfaces. Now what this graph really does is it provides a wonderful overview of the impact that the NIH has had on a very wide variety of disciplines. And I'm not even showing the whole piece of it here. We've sort of simplified it for the sake of creating a visual. And what I'd like to do is to highlight what BrainWorks' tool is capable of. In addition to just building a topic graph, I'd like to zoom in on one of the topics in this graph, specifically the neuroplasticity topic that you see right there. And if we take that and we kind of open it up, we might be interested in seeing, of the papers that were published on neuroplasticity, what are some of the other topics that co-occurred with those neuroplasticity papers? Because the co-occurrence of the topics could tell us things about trends in research. So for example, one of the simplest analyses we could do is we could take along the x-axis here the year, again 1980 to 2020. We could put along the y-axis the number of papers that exist within our archive. And we could split that number of papers according to some of the topical qualities of these neuroplasticity papers, such as whether they used humans or animals in the course of study. And what you'd see here, if you take your eyes and look from the left all the way to the right, is that animal studies were predominantly what were done for neuroplasticity research in earlier times, but as time has gone on, we've seen more work with humans in this domain. And we have taken the topics that we've imprinted upon these neuroscience papers, and we've hierarchically organized them. And what that allows us to do is say, if we want to drill into a particular topic and kind of explode it out, we have a capability to do that. Let's do that, for instance, with the animals topic represented by the orange bars here. We can take that animals topic and spread it out into various animals that sort of comprise that topic. So now what we have is the same plot that we showed on the last slide, but in addition to the humans in blue, instead of just having animals, we have the particular kind of animal that was used in neuroplasticity research at various points in time. And here what we see is that a slightly more nuanced story, that yes, while animals were used earlier and humans have been used a lot more in later times, the community switched from using rats more to using mice more in the course of experiments in neuroplasticity. Now, these kinds of conclusions are things that maybe a person who's not an expert at neuroplasticity or neuroplasticity research, it would take quite a lot of reading through papers to conclude something like this. And the goal of a tool like BrainWorks is to allow for the exploration of these sorts of associations so that more informed research decisions can get made. So if we come back now for a minute to this plot, I want you to imagine that the ambition of our tool is that for any topic that you see here and any topical interface that you see here, you could zoom in, get an idea of how things are changing over time, not only at the level of topics, but in the future at the level of the natural language text and its properties as well. But of course, a topic graph or any kind of research output graph, which is what this represents, isn't something that just exists. It's created by excellent researchers, the kind of researchers that you will be hearing from in some of the sessions later today. These are world leaders in their domain. And there might be a question of how do the interactions or collaborations between researchers in those domains impact the output of science and the kinds of things that we end up discovering. And BrainWorks also provides a way for us to understand the role of those authors in the creation of science. So for example, we might be interested in how collaborative projects impact author networks as well as their papers. And we might have an interest in using BrainWorks to answer a question like that. And so one thing that I did, because we were interested in this, was to take a population of around 1,500 researchers that were affiliated with 60 or so scientific labs, and to split them into an intervention and a control group. So 30 of those labs had received an award from the NIH to help foster additional collaborative efforts across labs, and then 30 of those labs, the other half, had received that same award, but at a later point in time. They got it 2019 or after. And so what we did was we said, okay, let's take these two groups who we know are probably similar in terms of their credentials. They received the same collaborative award at some point. Let's look at their activities 2 years before 2017. That's when the intervention group got it, and 2 years after, which is, we want to contrast what the output of the two groups were in these time periods to see what the impact of this collaborative award was on output. And what I'm showing you here in this plot is a network visualization that helps us understand some of the nuances of the answer. What I'd like to do for a minute is explain first how to interpret the nodes and the edges, and then we're going to talk a little more formally about what we concluded from the analysis. So what we have here, like any network plot, are nodes and edges. The nodes here correspond to individual authors. The size of the node indicates how much of an increase in the author's output there was over the 4-year period. And the color of the node indicates membership in a lab. So for example, you can see there's clusters of orange nodes, of green nodes, of blue nodes. That's because those people all work together in a scientific laboratory. You might also notice that there are edges, and the way to interpret those edges is that if there's any edge between authors, it means that over that 4-year period we were studying, there was an increase in collaboration. You might notice that some of those edges are orange, which means that they were collaborations across groups, different colored nodes, and then some of the edges are blue, which means that they were collaborations that increased within the group. Okay, so what I'm showing you here in this network is for that control group, the group that didn't receive the award in the period that we were thinking about, but received it later. This is how their collaborative network structure looks like. And what you can see here, if you kind of, just to highlight a couple things, is that there are several sort of islands that exist that I've highlighted where there was increased collaboration within the group, but then there were also some locations here where there were more collaborations between the groups. At the top, we see there's a small collaboration between 3 authors from 3 labs. There's a major collaboration between 2 groups, as indicated by that cluster of green and blue interacting. And then there is a little more minor collaboration between 2 groups that I'm showing there at the bottom. And this visualization allows us to sort of see the data at a level of nuance like that. Now, everything I showed you before was in a kind of control group. That was the group who didn't receive the award. If we take and do that exact same analysis, looking at changes in the output for these authors over the time period and changes in their collaborative relationships over the time period, what we see here is a graph that has some things in common, but then some important differences. And what I'd like to do is highlight first those differences. You can see here that in these 2 locations I've highlighted, there are these big kind of messy superclusters, I'm going to call them, with many different colors, and it's sort of a jumbled up mess. And now, while that may not look as pretty to your eye or to my eye, it's actually a really wonderful thing from a scientific collaboration perspective, because it means these authors were working together so closely that when we tried to sort of take this network and get people who were in clusters to be sort of as separate from each other as we couldn't easily detangle that group when we were constructing the graph. That bodes extremely well for what the impact of the collaborative award was in this case. And this is really meant to highlight that a tool that we're building allows us to do these kinds of investigations and to see nuance in the data that may not have been easy to do otherwise. So that sort of covers a quick overview of what we've done with the tool to date. And our next steps, what I will be working on between now and the end of the year, is the development of a user interface so that instead of seeing data like this in a presentation, you might be able to click through a website and play with some of these graphics yourself, do explorations on your own, and see what you come up with. Specifically, I'll be working on search functionality, graph representation, and group comparisons within the online tool over the coming months. And so with that, I would like to thank you for your attention. I'd like to really encourage you to attend and enjoy the excellent scientists that will be presenting later today, and also offer for you to reach out and contact me if you have interest in BrainWorks or ideas for what we might be able to do with the tool.